Everyone, I hope I'm live. I'm clearly and visible and audible to you people. Let me confirm in 1 minute. I will start the lecture ahead. Give me a minute to confirm it. Yeah, so I hope it is working. So I welcome you all for today's session. A very good morning to all of you. So I am Dr. Priyanka Sachdev here, and today I'm here to continue my series that I have started in pathology, super simplified pathology. So in pathology, there is a topic which we have started in the last lecture from the general pathology. So from general pathology, I have started cell adaptation, cell injury, and cell death, the first chapter in pathology. So this is the basics in general pathology. If you have understood this general pathology very well, it is very easy for you to understand complete systemic pathology. So the first chapter in general pathology is cell adaptation, cell death, and cell injury. So we have already covered first lecture on YouTube only. A day before yesterday, we have covered this chapter, that is cell adaptations, right? So I have already given the introduction. I will repeat it once. I will revise it once in the first 2-3 minutes of the lecture. Then I will come on today's short topic, that is cell injury, the various mechanisms of cell injury and morphology of the cell injury, right? We know that human body is made up of cells, right? From head to toe, we all are made up of cells. We are made up of multiple organs and organs are made up of cells. So we know that cell is the structural and functional unit. The cells are the structural and functional unit of all organs. And normally all cells are in homeostasis. What do you mean by homeostasis? Each cell of human body has a particular specific structure to perform the specific function. Just suppose the cells of the salivary gland, they do the function of secretion. The cells of the intestine, they do the function of absorption. Likewise, all the cells in human body have a particularly assigned specific function because they have a specific structure. Specific structure with specific function is known as homeostasis. So normally, this is a human cell, human body cell. So normally all cells are in homeostasis. But whenever any stress come on the cell can be the stress can be physiological, can be pathological. Can you see this is a stress? The stress can be physiological and stress can be pathological. You can understand, I guess, what is physiological stress. Physiological stress is not a disease. It is some physiology in human body like pregnancy, menstruation. These are not diseases, but this is not normal state. So that is physiological stress. So whenever any stress coming to this cell, three things happens one by one. The first thing the cell will do is cell adaptations. There are 5 types of adaptations which we have already covered in the last lecture. I will not repeat them. All adaptations are reversible. That is hypertrophy, hyperplasia, atrophy, metaplasia, and dysplasia. These are the 5 types of cell adaptations, right? All adaptations are reversible. Once the stress is removed, the adaptations are reversible. If the stress continues, it persists for a long time. The second thing happen is reversible cell injury. So second thing the cell will do is reversible cell injury. So after cell adaptation, now it is cell injury. Cell will get injured, but it is reversible. Again, it is reversible like adaptation. Once you remove the stress, cell will go to its original function. But if the stress still continues, if it is still continuing, the last thing that will happen is irreversible cell injury. Irreversible cell injury is cell death. The cell death is of 2 types, apoptosis and necrosis, right? Irreversible cell injury and cell death is one of the same thing, right? It is a synonym of each other. So what is the summary? What is the summary? The summary is that all cells are in homeostasis. Whenever any stress come on the cell, whether the stress is physiological or pathological, 3 things happen one by one in sequence. So what are the 3 things? The first thing, in response to stress, the cell will do is cell adaptations. The second thing, if the stress continues, it is cell injury. And the third thing, if the stress still continues, initially it is reversible, then it will be irreversible cell injury. Irreversible cell injury is known as cell death, and it is of 2 types, apoptosis and necrosis. It means that cell can die via 2 mechanisms, either by apoptosis or by necrosis. Who's going to decide cell will die by which mechanism? Apoptosis, necrosis are the 2 types of cell death. So who will decide that whether the cell will die by apoptosis or whether the cell will die by necrosis? We will come on that point in cell death or irreversible cell injury. Currently I am teaching you reversible cell injury, right? Now you can learn the 3, can I say cell adaptation, reversible cell injury and irreversible cell injury. These 3 things are progressive impairment of the cell normal homeostasis. So these 3 are the stages One by one, these are the stages. The first two are the reversible, the last is the irreversible. That is the thing. Have you got it? Now imagine the example. In the last lecture, I have given the same example. What is your self-study time? Usually students study for 6 to 8 hours daily. Normally, this is normal study time for a student. So that is your self-study time. So this is your homeostasis. So you daily study for 6 hours and this is your homeostasis. But now there is a stress. The stress is your exam. The exams are coming. So since tomorrow or few days after you have exam, there is a stress. So it is acting like a stress. Now you want to overcome this stress. If you don't overcome this stress, you will die. So you want to overcome this stress. So what you will do, you will do some structural changes inside you to gain the new homeostasis, right? So instead of 6 to 8 hours, you will study for 12 to 15 hours, right? So this is your adaptation. You are doing the adaptation in response to the stress. In response to the stress, you are doing the adaptation to bring a new homeostasis, to bring a new steady state, right? In order to overcome the stress. Otherwise, the cell will die. So same thing is happening with the cell also. The same thing is happening with the cell also. And once the exam are over, that is stress is removed. So again, you will study for 6 to 8 hours. Again, so again, you will come back to the homeostasis. So the adaptations are reversible. What if the exam continues? The cell injury, you will become ill. If you study daily for 15 hours, you will become ill. You will become sick, right? So that is cell injury will take place. So you can understand the same thing in this example. So in the last lecture, we have already covered the cell adaptations, right? Just a second. So in short, we have to study 3 chapters one by one. In the last lecture, we have already covered adaptation. In this lecture, I am going to cover injury. In the next lecture, I will cover cell death. So there are 3 portions in this chapter, right? Cell adaptations, we have covered the 5 type of adaptations: hypertrophy, hyperplasia, atrophy, metaplasia, dysplasia. I will not cover them again. You already know. If you have missed the lecture, please watch the recording. It is available on the YouTube. It is my lecture, cell adaptations, right? So it is available. So I have asked you to make this comparative table between the 5 type of adaptations, which their definition, diagram, mechanism, types, and morphology. So I guess this master table is ready with every student, and you can revise the cell adaptations just in 5 minutes by reading this table. All the information is given in this table. So please watch my last lecture, recording of last lecture, to know the details of this lecture. Currently, I am going to teach you cell injury, right? So let me start. After cell adaptation, the second thing which occurs is cell injury. As I have told you, cell injury is of 2 types, the reversible and irreversible. It is of 2 types. I will explain you both types of cell injury, reversible, irreversible. But the mechanism of both, whether reversible or irreversible cell injury, these are the 5 mechanisms of the cell injury. So let me explain you these mechanisms of cell injury. Give me a minute. Let me see if I can see your chat. Give me a minute. Yes, I can see your chat. So you can write down what, if you have any doubt, please write down in your chat. I can see the chat comment box, right? So there are 5 mechanisms of cell injury. I will explain you all 5 in simplified, super simplified diagrams and flowcharts, one by one. So the first mechanism is ATP depletion. Let me explain you one by one the 5 mechanisms of the cell injury. So the first mechanism is ATP depletion. The first mechanism of the cell injury is ATP depletion, right? So what is ATP depletion? Why the cell requires ATP? Okay, let me draw a diagram. So this is a cell. This is a cell. This is the nucleus of the cell. This is the DNA, double-stranded DNA of the cell. Can you see? This is the cell, nucleus of the cell, and double-stranded DNA of the cell. This is any cell of human body. This is the blood supply of this cell. All cells have blood supply. Imagine a situation if there is ischemia. What do you mean by ischemia? If there is obstruction in the blood vessel, so less blood is coming here to the cell. So cell is receiving less blood. That is ischemia is the injury. Ischemia is causing the injury. It is the etiology of the injury. So if cell is getting less blood, why cell requires blood? For oxygen. So for getting oxygen. So less blood means less oxygen. That is cell is in hypoxia. The cell is in hypoxia. Because of the ischemia, there is less blood. Less blood means less oxygen. Less oxygen means hypoxia. So less oxygen is present inside the cell. Why oxygen is required? Oxygen required for formation, for the synthesis of ATP. This oxygen goes inside the mitochondria of the cell, and in the mitochondria of the cell, oxidative phosphorylation takes place and ATP is synthesized in the mitochondria. Mitochondria is the powerhouse of the cell. It synthesizes ATP. Since less oxygen is going in the mitochondria, less ATP or no ATP are synthesized. So it is known as ATP depletion. This mechanism is known as ATP depletion. This is a stress. This is a stress. So what is the stress to the cell? The stress to the cell is ATP depletion. Why ATP depletion is there? Because mitochondria is not forming ATP. Why mitochondria is not forming ATP? ATP is formed from oxygen. So oxygen is absent or it is less. Why oxygen is less? Oxygen is present in blood. So this cell is getting less blood because of ischemia. So ischemia is the event. So what is ischemia? Ischemia is leading to less blood in the cell. Less blood means less oxygen in the cell. Less oxygen means less ATP or no ATP in the cell. This is known as ATP depletion. Now you should ask me, ma'am, because of absence of ATP, what are the problems in the cell? There are three problems in the cell because of ATP depletion. Because of ATP depletion, the cell will face three type of problems. What are the three type of problems? I will explain you all one by one. Okay, let me draw another diagram. Imagine this is a cell. This is the nucleus of the cell. This is the DNA of the cell. And this is the mitochondria. Can you see? This is the mitochondria of the cell. In the mitochondria, no ATP is synthesized. So ATP is less or absent. This is known as ATP depletion. ATP is depleted. The reason we know, the reason is ischemia. Because of ischemia or hypoxia, there is less ATP. Now there are three stories I am going to tell you one by one. One by one I will tell you three stories. What will happen in the cell? On the surface of the cell, there is a sodium pump. There is sodium potassium pump present on the surface of the cell. This is sodium potassium pump. So normally this pump operates in presence of ATP. If ATP is present, then only this pump is operating. Otherwise, this pump is not operating. So when ATP is present in this cell, this pump takes 3 sodium out, and it takes 2 potassium in. So this is sodium potassium pump. Normally it happens. Sodium goes out and in exchange of it, potassium comes in. So that is sodium potassium pump, but the condition is that it functions in presence of ATP. Now I am saying that ATP is absent. ATP is absent. Since ischemia is there, hypoxia is there. So ATP is absent. Since ATP is absent, so this function, the sodium potassium pump will not work. So what will happen if it will not work? Sodium will not go out and potassium will not come in. So sodium will remain inside only and potassium will remain outside only. Sodium will not go out. Now sodium is an osmotic carrier of water. Sodium के पीछे पीछे पानी जाता है. Wherever sodium goes, water goes. Wherever sodium goes, water goes. It is a golden rule. If sodium is not going out, water is not going out. So water is causing accumulation in the cell. So this water, because sodium is not going out, water is not going out, it is accumulating in the cell and it will cause cell swelling. It will cause the cell will swell up. The cell will swell up. After a point, it will burst and die. So cell will swell up. Number one, it is causing cell injury. So cell injury is due to the swelling of the cell. Along with swelling of the cell, one more thing is there. Can you see the cell will swell? Along with the swell, the blebs are formed. On the surface of the cell, blebs. Can you see? These are the blebs. The blebs are formed on the surface of the cell and the cell will swell. These two things happen because of this thing. Because of sodium potassium pump, which is non-functional. Number one. Now, the second thing. What will happen now? If ATP is absent, so cell try to make ATP from some other source. Normally, ATP is formed in the mitochondria from oxygen. It is known as oxidative phosphorylation, right? Now, if oxygen is absent because of hypoxia, cell will try to make ATP from some other mechanism that is known as anaerobic. In absence of oxygen, anaerobic glycolysis. The mechanism is anaerobic glycolysis. So you have studied in biochemistry that is in aerobic glycolysis. From one molecule of oxygen, 36 molecules of ATP are formed. But in anaerobic, from one molecule of oxygen, only 2 ATP are formed. Okay, so here 2 ATP will be formed. But what is the end product of anaerobic glycolysis? In anaerobic glycolysis, the end product is lactic acid. So it will lead to formation of lactic acid. So lactic acid is formed in the cytoplasm and inside the cell. So this lactic acid will cause acidic pH of the cell. It is an acid, so it will reduce the pH of the cell. And reduced pH, the acid will go in the nucleus and cause clumping of the nucleus. So the nucleus is clumped. Clumping of the nucleus will take place because of acidic pH. Now imagine a cell which is swelled up, the surface is showing blebs and the nucleus is clumped. What is the reason for swelling? Sodium potassium pump not operating. What is the reason for blebs? The same reason, sodium potassium pump not operating. And what is the reason for clumping of nucleus? The reason for clumping of nucleus is lactic acid formation, which is leading to acidic pH of the cell. Give me a thumbs up if you got it. That is the second story. The third and the last story, inside the cell we have endoplasmic reticulum. You know, inside the cell, endoplasmic reticulum, many endoplasmic reticulum are present. Endoplasmic reticulum are of two types, the rough and the smooth. Does anyone know what is the difference between the rough and the smooth? Rough endoplasmic reticulum are those on which ribosomes are present on their surface. If ribosomes are present on the surface of the endoplasmic reticulum, it is known as rough. If ribosome is not present, it is known as smooth. For binding of ribosome with the endoplasmic reticulum, ATP is required. ATP is required. And inside ribosome, protein synthesis takes place. We know protein synthesis takes place in the ribosome 30S, 50S unit, right? So protein synthesis. So if ATP is absent, the ribosomes will detach. It will detach from the endoplasmic reticulum. So no protein formation take place. So cell will be deployed of protein. All enzymes are made up of protein. Cell structure is made up of protein. Cell membrane is made up of protein. So everything will be depleted. So cell ultimately will die. So this is the story. Have you got it? Have you got the complete story? So what are the three stories? You yourself tell. Let me see if I can see your chat, yes or no. Give me a minute. Just a minute. So have you got the three stories? ATP depletion. What is happening in ATP depletion? Any one of you. What is actually happening in ATP depletion? During ATP depletion, so this diagram is from Robbins. Can you see ATP is depleted? So during ATP depletion, three things are happening. Number one, sodium potassium pump is not operating. Because for operation of this pump, we know why ATP depletion occurs. Start from the beginning. So there is ischemia. Can you see a blood vessel? Beautiful diagram from the Robbins. So can you see a blood vessel? Yes or no? Can you see it? Vasi, Sohil, yes. So you can see a blood vessel. So there is ischemia in the blood vessel. Ischemia means less blood. Less blood means less oxygen. Less oxygen means hypoxia. So in hypoxia, can you see this is mitochondria of the cell? It is mitochondria. Inside the mitochondria, there is reduced oxidative phosphorylation because oxygen is absent. So less ATP will be formed or no ATP will be formed. Yes, so less or no ATP will be formed. In absence of ATP, three things will happen one by one inside the cell. Now see the diagram of the cell, it is given here. All the things I will show you in this cell, right? So sodium potassium pump will not function. If sodium potassium pump, why it will not function? For functionality, it requires ATP. Since ATP is absent, it will not function. So it will not function, so sodium will not go out. So there is influx of sodium. Along with sodium, it is the water which will remain inside the cell. And potassium ka efflux ho jayega. So potassium ka efflux, sodium and water ka influx. It will cause cell swelling. Number one, cell swelling, endoplasmic reticulum swelling, and blebs. Can you see the blebs formation? Appreciate the blebs formation. Here is bleb formation. Appreciate this bleb. So on the surface of the cell, it is bleb formation due to this. And the cell is swelled up. Please appreciate the cell is swelled up. That is the first thing. The second thing, now since ATP is absent, cell will try to make ATP by some other mechanism. So if oxidative is absent, oxidative phosphorylation cannot take place because oxygen is absent, right? So cell will try to make ATP from anaerobic glycolysis. Anaerobic glycolysis, so 2 ATP are formed, okay. But the end product of glycolysis is lactic acid. So inside this cell, lactic acid will be formed in the cytoplasm. This lactic acid will reduce the pH of the cell. Since it is an acid, it is not a base, it is an acid. So it will reduce the pH of the cell. And reduced pH, the acid will cause clumping of the nucleus. See the nucleus. Please appreciate, everyone appreciate the nucleus of the cell. It is clumped. If you can appreciate, the clumps are present in the nucleus. Can you see? The nucleus is clumped. So imagine a cell which is swelled. The surface is showing blebs. The cell organelles, mitochondria, endoplasmic reticulum, they are also swelled. And the nucleus is clumped. That is, cell injury is going to take place one by one, right? Now the third thing which is going to take place here, ribosomes will detach from endoplasmic reticulum. Because for binding ribosome with endoplasmic reticulum, it requires ATP. Since ATP is absent, so ribosome will leave the endoplasmic reticulum. Since ribosome will detach from the endoplasmic reticulum, protein synthesis do not take place inside the ribosome. Protein synthesis do not take place, so no enzyme formation, no cell membrane formation, no cell structure formation, all things will be absent in the cell. So imagine a cell which is swelled. The cell surface is showing the blebs. The nucleus is showing the clumping. There are no enzymes. There is no cell membrane. It is showing the detachment of the cell membrane, and cell structural components are absent. Give me a thumbs up. These are the various mechanisms of cell injury due to ATP depletion. So I will teach you 5 mechanisms. This is the first one. I want thumbs up from all of you. If you have any doubt, please ask it. That is ATP depletion. ATP depletion. Otherwise, give me a thumbs up. So the first mechanism, ATP depletion, is done. You can see ATP depletion. The diagram from the Robbins is in front of you. This is a diagram from Robbins. I hope you got this diagram, right? This is sodium potassium pump. Can you see the cell membrane here in this diagram? This is a cell membrane. On the cell membrane, can you see this is sodium potassium pump? Can you see it function in presence of ATP? In presence of ATP, see the arrows. See how arrows are working. Everyone see the arrows. Okay. It is taking sodium out. It is taking 3 sodium out, extracellular, and it is taking 2 potassium in, normally in presence of ATP. But if ATP is absent, it will not function. So sodium will remain inside only and potassium will remain outside only, intracellular and extracellular. You got it? You got it? So, and this is the diagram of the ribosomes. Normally ribosomes, this is the 2 units of the ribosomes. This is 50S and 30S, the 2 units of the ribosome. Now it does protein synthesis only when it is attached to endoplasmic reticulum. If it is detached from endoplasmic reticulum, protein synthesis will not take place. And attachment to endoplasmic reticulum requires ATP. So if ATP is absent, it will detach and no protein synthesis take place. Have you got it? So please write the 3 flowcharts. The 3 flowcharts in your exam. One by one, please tell me the 3 flowcharts. So due to ischemia, there is no oxygen. Because of absence of oxygen, oxidative phosphorylation in the mitochondria is reduced. Since oxidative phosphorylation is reduced, there is reduced ATP formation. Because of reduced ATP formation, there is reduced sodium potassium pump action. So sodium and water will enter the cell and potassium will exit from the cell. Since there is more water inside the cell will dilate, the cell will swell and blebs will be formed. Give me a thumbs up. That is the first thing. That is this one. This one I have explained. The second, in absence of oxygen, cell will try to form ATP from anaerobic glycolysis. So anaerobic glycolysis may glycogen will be lost and lactic acid is the end product. More and more lactic acid is formed. That will create acidic pH of the cell. Because of acidic pH, the nucleus will show chromatin. The nuclear material will show the chromatin clumping of the nucleus, the nuclear material chromatin. Give me a thumbs up. That is the second thing. The third and the last thing, because of decreased ATP, the rough endoplasmic reticulum will lose the ribosome. The ribosome get detached from it. So if ribosomes is getting detached from it, there is no protein formation. Since protein formation is absent, so no enzyme, no cytoskeleton, no cell membrane. Everything will be absent in the cell. So this is the third thing. So ultimately this cell is formed. Give me a thumbs up. So the first mechanism, Sohil, you are asking a very good question that these cell injuries are reversible. It is an overlap. So ATP depletion is the cause for reversible as well as irreversible cell injury. Now, as per your question, what things I am telling you, that is clumping of chromatin, the bleb formation, the cell swelling, are all these reversible? Yes. Initially, yes, your answer is right. Your question is right. Initially, these all are reversible, but later on, they will convert into irreversible. I will come on that point. Let me teach you the 5 mechanisms. Then I will teach you the morphology of the cell injury, in which I will definitely answer your question with detail. Have you got it, Sohil? Should I proceed ahead? So coming on the next mechanism, the second mechanism. Can you see the second mechanism? The name of the second mechanism is mitochondrial damage. So let me teach you mitochondrial damage, right? The second mechanism of cell injury is mitochondrial damage. Oh my God, just a second. Okay. Just a second, give me a minute. Yeah. So here I am. So it is the second mechanism I'm teaching you is mitochondrial damage. So in mitochondrial damage, you know mitochondria is the powerhouse of the cell. In mitochondria, ATP formation takes place. Mitochondria is the powerhouse of the cell. So because, now imagine any injurious agent. Now initially in the first mechanism, the injurious agent was ischemia. Due to ischemia, the event was starting. Due to ischemia, there is hypoxia. Due to hypoxia, ATP depletion was taking place because of reduced oxidative phosphorylation. Here, the injurious agent can be anything. It is not always ischemia. It can be physical, biological, chemical, some microorganisms, some bacteria, anything which is causing mitochondrial damage. So anything who is causing, it can be ischemia also. Anything who is causing mitochondrial damage, it will result in two things. It will result in two things. So there are two flowcharts in mitochondrial damage, right? There are two flowcharts here. So listen, normally inside the mitochondria, so this is a cell. Inside the cell, this is the nucleus. I'm interested in mitochondria. Let me draw mitochondria here. Normally, listen, oxidative phosphorylation takes place inside mitochondria. Oxidative normal oxidative phosphorylation takes place inside the mitochondria, because of which ATP is formed. Right. Now this mitochondria is damaged. Imagine a situation in which there is damage to the mitochondria. The mitochondria is damaged. Who is causing the damage? Anything can cause the damage. Here injury can be anything. It is not always ischemia. It can be physical injury, it can be chemical injury to the cell, it can be biological injury. By biology, I mean it can be bacteria, virus, fungus, parasite, any microorganism injury. So mitochondria of the cell is damaged. If mitochondria is damaged, two things will happen. Normally, normal oxidative phosphorylation takes place now. So here normal oxidative phosphorylation will not take place. Normal will not take place, so no ATP formation. If normal oxidative phosphorylation takes place, so ATP formation takes place. But normal oxidative phosphorylation will not take place, so ATP depletion. एक तो होगा ATP depletion. Second, abnormal oxidative phosphorylation takes place. Abnormal oxidative phosphorylation will take place because of the damage. Because of normally in normal oxidative phosphorylation, oxygen enters the mitochondria and get converted into ATP. Oxygen get converted into ATP. But in abnormal oxidative phosphorylation, which takes place in damaged mitochondria, oxygen get converted into free radicals. So here free radicals are formed. The free radicals will cause damage to the cell. There are three types of free radicals. What are the three types of free radicals? Can anyone tell me? Can you enumerate the three types of free radicals? It is superoxide formation, superoxide ion formation, number one, the first one. Second is hydroxyl ion, hydrox O2 negative, OH negative, and H2O2, hydrogen peroxide. So these are the three basically these three free radicals are formed in the cytoplasm of the cell, and they will cause complete cell damage. They will damage the cell membrane, they will damage the cytoplasm, cytoplasmic proteins, and they will damage the nucleus. Everything of the cell will be damaged, and it will ultimately result in irreversible cell injury. Give me a thumbs up. So that is the mitochondrial damage. So you can see that is the second thing. One more thing, the third thing will also happen here. Okay, I would like to draw a diagram again. Okay, so let me draw a mitochondria. This is the mitochondria. These are the two layers, the two membranes of the mitochondria. Mitochondria is a double layered cell organelle, inner membrane, outer membrane, right? So normally in mitochondria, normal oxidative phosphorylation takes place, which results in ATP formation. This is the normal function of mitochondria in me, in you, in everyone, right? But due to any reason, if this is damaged, if this mitochondria is damaged due to any reason, physical, chemical, biological, so three things will happen. Number one, normal oxidative phosphorylation will not take place because it is damaged. So ATP will not form. In normal, oxygen get converted into ATP, so this conversion do not take place and no ATP formed. So what the oxygen will get converted now? In the damaged mitochondria, not in healthy. If mitochondria is healthy, this takes place. Damaged में abnormal oxidative phosphorylation takes place. What is abnormal? Normally oxygen get converted into ATP, but now oxygen get converted into free radical. So three types of free radicals are formed. You know the name? It is superoxide ion, hydrogen peroxide, and hydroxyl ion, OH negative. They will cause the cell damage. They will cause cytoplasmic damage, cell membrane damage, cytoplasmic damage, and nuclear damage. So cell is damaged. Number two. So, and the third thing, between the two membranes of the mitochondria, listen this one, listen this one. Between the two membranes of the mitochondria, there is a protein. The name of the protein is cytochrome C. Normally, it is present between the two layers of the mitochondria. It never comes out. Now, when mitochondria is damaged, this cytochrome C will come out. Cytochrome C will come out of the mitochondria and it come in the cytoplasm. From the mitochondria, it comes out in the fluid that is in the cytoplasm. Once it comes in the cytoplasm, if you know the mechanism of apoptosis, it will cause apoptosis of the cell. So apoptosis is ultimately irreversible cell injury, it is cell death. Have you got it? Have you got it? So three things will happen due to mitochondrial damage. Number one, normal oxidative phosphorylation will not take place. So ATP depletion. ATP depletion further results in those three things. You already know in the first mechanism. All the five mechanisms are connected with each other. So ATP depletion से वही तीन चीजें होंगी. What three things? Sodium potassium pump will not operate, cell will swell. Number one, ribosomes will detach from the endoplasmic reticulum, so protein synthesis will not take place. And anaerobic glycolysis will take place, that will result in lactic acid formation, that will result in nucleus clumping. So ये सब तो होगा ही होगा. It is, I am assuming that you know it. So normal oxidative phosphorylation will not take place and abnormal will take place. If normal will not take place, no ATP formation. If abnormal will take place, so free radical formation take place. So normally oxygen get converted to ATP, it is not taking place. Now oxygen is getting converted into free radical. So free radicals which are formed, they will cause three damage: cell membrane damage, cytoplasm damage, nucleus damage. Give me a thumbs up. Have you got it? And the third thing, between the two membranes of the mitochondria, a protein is present which is known as cytochrome C. Normally cytochrome C never comes out of the cell. Cytochrome C comes out of the cell if cytochrome C comes out of the cell in mitochondrial damage, it will result in apoptosis. Apoptosis is cell death. Actually, it is cell suicide. Cell will commit suicide. When cytochrome C will come out, cell will commit the suicide. Cell will commit the suicide. That is a type of cell death. I will tell you the details of apoptosis, Dr. Sri Rathod, in my next lecture. I will tell you all the complete mechanism of the apoptosis. I request you to attend my next lecture after cell injury, that is apoptosis, in which I will give you the detailed description what actually the cytochrome C doing in apoptosis. So step by step. So here that much is sufficient. Have you got it? Everyone got it? Should I proceed? So that is the second way. So see, the diagram is from Robbins. You can see the diagram, the beautiful diagram from the Robbins, right? So mitochondrial damage. If this mitochondria, you can see a mitochondria if it is damaged. If it is damaged due to any reason, it can be physical, chemical, biological, it can be any reason, toxins, radiation, right? Mitochondrial damage is there. So normal oxidative phosphorylation will not take place, so no ATP formation, number one. And abnormal will take place, so it will result in free radical formation. Free radical का दूसरा नाम है, other name of free radical is ROS. It is reactive oxygen species. What are free radicals? As I have told you, there are three free radicals. What are three free radicals? Superoxide ion, hydroxyl ion, and hydrogen peroxide. These three are the free radicals. These are known as reactive oxygen species, ROS. So free radicals are nothing. These are ROS. It is written in the books. It is known as ROS only. Reactive oxygen species, because they are formed from oxygen. If mitochondria is damaged, oxygen get converted into these three. So that is reactive oxygen species. Have you got it? Have you got it? So I will come on the cellular aging. It is not a lecture on cellular aging, Ayushi. So definitely I will cover the mechanism of cellular aging in my next lecture. After apoptosis, necrosis, there is a lecture of cellular aging. So I will tell you the details there, that is telomerase theory and everything there, Ayushi. But here I am teaching you the various mechanisms of the cell injury. It is not a mechanism of cell injury in aging, if you are saying that, right? So in aging, the main mechanism will be the telomerase theory, that I will tell you there. Anyways, should I proceed ahead? So that is the two things. Normal oxidative phosphorylation will not take place, so no ATP formation. Abnormal will take place, so reactive oxygen species or free radical formation will take place. And the third thing, can you see the two membranes? See the first membrane and the second membrane. Between the two membranes of the mitochondria, this is cytochrome C, the red color molecule. This cytochrome C is leaked out. So leakage of cytochrome C. Name of the protein here is cytochrome C. It is leaking out and it is causing apoptosis of the cell. Everyone gave me a thumbs up. Everyone give me a thumbs up. So please write down three things one by one. In mitochondrial damage, please draw the three flowcharts. What are the three flowcharts? This is the first flowchart. If mitochondrial damage is taking place, so normal oxidative phosphorylation do not take place, right? So there is no formation of ATP, and loss of ATP will result in that three things, you know. The second thing, mitochondrial damage will lead to abnormal oxidative phosphorylation. So oxygen will get converted into free radicals. So ROS are formed, reactive oxygen species, that are known as free radical. They are formed and they have deleterious effect on the cell. They will cause cytoplasmic damage, they will cause cell membrane damage, they will cause nucleus damage. In short, they have deleterious effect on the cell. And the third thing, the last thing, the mitochondrial sequester, that is cytochrome C, which is normally present between outer and inner membrane, that will leak out. Due to mitochondrial damage, it will leak out. It will cause leaked out. Increased permeability means leakage ho jayega cytoplasm mein. So there is leakage of cytochrome C in the cytoplasm, and that will lead to apoptosis. So I am done. I am done. So this is the summary in front of you of the second mechanism of cell injury. Mitochondrial injury will result in three things. Number one, ATP depletion, because normal oxidative phosphorylation do not take place. Number two, formation of ROS, that is because abnormal oxidative phosphorylation takes place. And number three, apoptosis. It is due to leakage of cytochrome C in the cytoplasm. Everyone give me a thumbs up. Everyone give me a thumbs up. So this is the summary of second mechanism. Let me revise you two mechanism, then I will come on the third mechanism. Till now I taught you two mechanism. So three effects of the first mechanism and three effects of the second mechanism. So please everyone draw the flow charts. The flow charts are important. The topic will be super simplified for you, as my series name is Super Simplified Pathology. So I will make the entire Robbins super simplified for you. After watching my videos, listening my videos, reading Robbins is like cakewalk. It is like cakewalk. So after today only, after watching my lecture, please read it in from Robbins only. So you'll just flip the page. Yeah, I know this, and you will get it very easily, right? So that is the thing. That is the thing. So let me revise you the first two mechanism and then I will come on the third mechanism. That is loss of calcium homeostasis. So, or you want me to take third also, then we will revise the three together. Okay, I'm taking the third also. Loss of calcium homeostasis. The third mechanism of the cell injury. Loss of calcium homeostasis. What do you mean by that? What do you mean by that? Okay, listen. In cell, where is the calcium present? Does calcium present in the cell? This is the cell. This is the nucleus of the cell. This is the DNA of the cell. This is a human cell. My cell. Any normal human being, any cell from head to toe. Where is the calcium present in the cell? Normally, 99% of the calcium present extracellular. It is not present intracellular. Only 1% of the calcium is present intracellular. That too, that 1% also is not present in cytoplasm. So where does it present? It is not present free in the cytoplasm. It is present either in mitochondria or is present in endoplasmic reticulum. Inside that, only 1% is present. 1% or very small amount of calcium is present. Maximum calcium is extracellular. Minute amount is present intracellular also, that too not free. It is not present freely in the cytoplasm. It is present inside two cell organelles. What are the two cell organelles? It is mitochondria and endoplasmic reticulum, inside which this calcium is present. Give me a thumbs up if you got this point. Now imagine any injurious agent. Again, it can be physical, chemical, biological, toxin, radiation, bacteria, anything causing injury to this cell. Anything causing injury to this cell. Due to cell injury, two things will happen. Number one, extracellular calcium will come in the cytoplasm. And number two, the intracellular concentration of the calcium will also leak the calcium in the cytoplasm. So in the cytoplasm, transiently, there is abrupt increase in concentration of the calcium. It is coming from extracellular also, intracellular also. Give me a thumbs up. So transient increase in concentration of calcium in the cytoplasm. So what will happen now? Calcium बढ़ गया cytoplasm में, तो उससे क्या problem है? What is the problem? In the cytoplasm, 4 enzymes are present. They are inactive and calcium is the activator for them. So all the 4 enzymes will get activated. What is the name of those 4 enzymes? The first enzyme, the name of the first enzyme is ATPase. ATPase. It will get activated, and what it will do on activity? On activating, it will cause the degradation of ATP. So ATP are lost. The second is DNase. What it will get activated because of calcium. So what does it do? It will degrade the DNA. The third is the phospholipase. So it will get activated because of calcium, and what it will do? It will do the degradation of the phospholipid. Phospholipid is the cell membrane. So cell membrane is lost. And the fourth one is ek aur hai. I will tell you. So all the enzymes will get activated. Have you got it? Let me show you the same diagram from the Robbins. So this is a diagram. I will show you all the diagrams standardly from Robbins only. So Robbins is it is the Bible of pathology. So I follow only one book in pathology, that is Robbins, and I will super simplify the Robbins for you, right? So on YouTube only, I will launch the entire series of the complete pathology one by one, chapter by chapter, and the complete general hematology and systemic pathology from Robbins. I will super simplify for you. Are you liking the way of teaching? Are you getting the concept? Okay. See in this diagram, can you see a cell? See, maximum calcium is extracellular. This is extracellular, it is 99%. See the cell. Inside the cell, it is present at two positions. Mitochondria, it is present inside mitochondria, and it is present inside endoplasmic reticulum. This is only 1%. But when cell injury is taking place, can you see this is the injurious agent, because of which two things are taking place. All the calcium is coming in the cytoplasm. Cytoplasm, it is coming from extracellular source also, and it is coming from intracellular source also. Give me a thumbs up. So there is transient increase of the cytoplasmic calcium suddenly. Suddenly the calcium in the cytoplasm increases transiently. So it causes activation of 4 enzymes. Name the 4 enzymes. What are the 4 enzymes? Number 4, ATPase. It will cause depletion of ATP. ATP depletion, again same 3 things. I am not repeating, you are knowing it. Sodium potassium pump inactivation, ribosome detachment from endoplasmic reticulum, and anaerobic glycolysis leading to lactic acid formation and decreased pH. So you know the 3 consequences of ATP depletion that will happen, right? Number 1. Number 2, phospholipase, it will cause degradation of the phospholipid, so cell membrane is lost. Number 3, endonuclease or DNase, that will cause degradation of the nucleus. And number 4, protease, that will cause degradation of various proteins. So you found it, the 4 enzymes will cause complete degradation of the cell. So imagine a cell in which cell membrane is lost, the protein are lost, the nucleus is lost, and ATP is lost. Imagine a cell. So that is the various mechanisms of cell injury inside the cell. Give me a thumbs up. So see the flow chart. The maximum calcium is present intracellularly. Maximum calcium is present extracellularly. Intracellular concentration is very low, only 1% as compared to extracellular. We know it. And intracellularly also, it is not present in the cell cytoplasm. It is present in mitochondria and in endoplasmic reticulum. Have you got it? Have you got it? So we can see it. Now, this is the flow chart. So due to injury, any injury, the cytoplasmic calcium will increase because of two things. What are the two things that will increase cytoplasmic calcium? Because of influx of calcium, the extracellular calcium is coming in, and release of calcium from the intracellular stores. That is mitochondria and endoplasmic reticulum is also releasing calcium. So calcium in the cytoplasm is coming extracellularly as well as intracellularly. This increase in calcium causes activation of four enzymes. Phospholipase, which will cause cell membrane damage. Protease, which will cause damage to the protein. Nuclease, that is DNase, that will cause damage to the DNA. And ATPase, that will cause damage to the ATP depletion. So cell damage will take place. Give me a thumbs up. Give me a thumbs up. So till now, we have done the three mechanisms of cell injury. I hope you don't have any doubts on this. So ATP depletion, there are three fates of ATP depletion. Mitochondrial damage, there are three fates of mitochondrial damage. And loss of calcium homeostasis, it results in activation of four enzymes. You know the name of the four enzymes: phospholipase, ATPase, endonuclease, and protease. So that is the activation of these enzymes that will cause cell injury. Now I will come on the fourth and the fifth mechanism. Till now, do you have any doubt? Anyone having any doubt? Anyone? What Ayushi is saying? You are a second prof student. You can suggest me which book I should, Robbins or Hirschman. So Ayushi, you two books you have mentioned, Robbins or Hirschman. So I prefer, you are a second prof student. So you have to give two exams. First your university exams right now, and after 3-4 years you have to give next. So till you will complete your final year, there will be next. Then NEET PG aims, everything is replaced by next from the next year, right? So you have to give next also. So if you want to crack next with a good rank, with a single digit rank, so you have to read Robbins. Without reading Robbins, you cannot crack next, right? But for university exam, Hirschman is also sufficient. So if you want to clear your university exam with good marks, with distinction, because what is the problem now in Robbins, the little bit the language is difficult. So Robbins may language is little bit difficult. Big paragraphs are there, points are not there, big paragraph, and everything is at genetic level, explained with molecular basis and genetic level. So students found it little bit difficult to read. Number one, it takes more time to read. Number one, and number two, retention is less. Retention is less. So once you read the Robbins, today you have finished 10 pages of Robbins or 15 days. But tomorrow I will ask you what is the summary of last day reading. So you will say, I don't remember anything. That is the point. And Hirschman is in points, 1, 2, 3, 4, 5, 6. Like, so retention is better in Hirschman. So for university exams, your Hirschman is better. But for next, you have to read Robbins. Now, what is the way to read Robbins? Watch the lectures of the educators of pathology who give the lectures based on Robbins. I am one of them. Many other educators are also doing that. So Robbins series करके बहुत सारे lectures आते हैं. Most of the lectures are from Robbins. I prefer Hirschman also. Few of the diagrams are from Hirschman also, you can see. But most of the theory part will be from Robbins. I have converted Robbins into points. Like Hirschman, point by point, flowchart by flowchart. All my flowcharts are from Robbins only. It is not paragraph. Now you read Robbins, it will be super simplified for you. Have you got your answer, Ayushi? Done. Should I proceed ahead? Should I proceed ahead? Okay, let me see if I can see your chat. Give me a minute. So I will continue with the next mechanism. Yeah, I can see. So I will continue with the next mechanism of the cell injury. The next mechanism, the fourth mechanism of the cell injury is defect in cell membrane. It is very easy one. It is very easy one. The defect in the cell membrane, the cell permeability. So membrane, not cell, it is membrane permeability. So actually due to the cell injury, now, okay, this is a cell. Let me draw the diagram of a cell. This is the nucleus of the cell. So this is cell membrane. Cell is bounded by cell membrane and nucleus is bounded by nuclear membrane. So cell membrane, nuclear membrane, and this is mitochondria. Mitochondria is bounded by mitochondrial membrane. I have drawn three membranes. In this diagram, you can see three membranes. The first is the cell membrane, the second is the nuclear membrane, and third is the mitochondrial membrane. Mitochondrial membrane, plasma membrane, and cell membrane. Okay, the third is the not nuclear membrane, it is lysosomal membrane. You know, inside the cell, we have lysosomes. We have lysosomes inside the cell. So this is lysosomal membrane. So the injury can cause damage to any membrane. If the damage is caused to the cell membrane, you imagine what will happen. The cell membrane will have gaps. The cell membrane will have defects and all the plasma will leak out. If the plasma will leak out, cytoplasm will leak out, the cell will die. So if the cell membrane, plasma membrane is damaged, the cell will die because of leakage of the vitals. Number one. If the mitochondrial damage will take place, I have already taught you mitochondrial membrane damage होगा तो क्या क्या होगा. Mitochondrial damage में the three consequences. And if the lysosomal membrane is damaged, what is lysosome? What do you mean by lysosome? Inside the cell, let me draw a cell. This is a cell. This is the nucleus of the cell. We have lysosomes inside the cell. What are lysosomes? Lysosomes are known as suicide bag. Suicide bag. Normally, lysosomes are intact. Their cell membrane is not defective. But if they rupture, if there is a defect in their cell membrane, the cell membrane have holes. So, lysosome contains enzymes inside them. These enzymes will come in the cytoplasm, the same enzymes. Endonuclease, phospholipase, and protease, the various enzymes that will cause degradation of the ATP, the nucleus, the phospholipid, the protein, everything. These enzymes are active and normally they are present inside the bag. So that's why lysosome is known as suicide bag. Once the lysosome is ruptured, the cell will commit suicide. So all these enzymes will degrade the cell entirely and cell will die. That's why it is known as suicide bag. Give me a thumbs up. Everyone, give me a thumbs up. Everyone give me a thumbs up. Because of calcium, it's already damaged. So what you are asking, Basi? Are you asking, do you want to ask anything? Yes. So have you got it? So that is the membrane damage. So three type of cell membrane damage takes place. Number one, mitochondrial damage, that I have already taught you. Due to mitochondrial damage, the three things happen. You already know. Normal oxidative phosphorylation do not take place, so no ATP formation. Abnormal take place, so ROS formation and release of cytochrome C that will lead to apoptosis. Number one, right? Plasma membrane damage. If the plasma membrane is lost, the fluid will come out. There is leakage of the fluid and the cell will die. There is loss of the cellular contents and cell will die. And lysosomal membrane damage. So all the enzymes, DNAs, RNAs, protease, phospholipase, glucosidase, cathepsins, all will leak into the cytoplasm and they will degrade DNA, RNA, protein, glycogen, everything. So cell will die. Cell will die. So that is lysosomal membrane damage. Have you got it? So can you see this is a lysosome? This is a lysosome and it contains many enzymes inside it. And once it got damaged, it got ruptured. Can you see the defects inside it? Once it got ruptured, the defects in the cell membrane, so enzymes will leak out and that will cause cell damage. Give me a thumbs up. So the fourth mechanism is defect in the cell membrane. So till now the concepts are clear to you. And the last and the fifth one, but not the least, is free radical injury. Free radical injury. So the fifth mechanism of cell injury, which is most important, is free radical injury. Everyone give me a thumbs up. Should I start free radical injury? The last one, the free radical injury, the fifth mechanism of the cell injury is free radical injury, right? So free radical injury, what is free radical? What do you mean by free radical? As I've told you, free radical are also known as reactive oxygen species, ROS. One and the same thing, free radical and ROS. So anything which is having a single unpaired electron in the outermost orbit is known as free radical. That is the chemical definition of free radical. In chemistry, you may have studied orbit. So outermost orbit means single free electron, unpaired electron. Electrons are present in pairs, no? So if single unpaired free electron is present in the outermost orbit of any molecule, that molecule is known as free radical because it is highly reactive. Because the unpaired electron is present, it is very reactive and it will attack the protein, lipid, carbohydrate, nucleic acid of the cell and damage everything. So these are reactive oxygen species or free radicals. They have single free electron in their outermost orbit and they cause damage to everything. They cause damage to protein, they cause damage to lipid, carbohydrate, nucleic acid of the cell. Give me a thumbs up if you got it. Give me where they are generated. They are generated in the mitochondria. The free radicals are generated in the mitochondria. All the free radicals are generated in the mitochondria. Listen, okay, inside the mitochondria, as I have told you, let me draw a mitochondria. So this is a cell. Inside the cell, this is the mitochondria. In the mitochondria, oxidative phosphorylation takes place, as you know. What do you mean by oxidative phosphorylation? What do you mean by oxidative phosphorylation? Does anyone know? In biochemistry, you have studied it in detail now. Tell me, what do you mean by oxidative phosphorylation? Oxidative phosphorylation hota kya hai? What do you mean by that? So in oxidative phosphorylation, oxygen enters in mitochondria. It takes place on inner membrane of the mitochondria. There are two membranes now. Inner membrane pe oxygen enters inside. So oxygen ultimately get converted into water. And during this process, ATP are released. At various steps, many ATPs are formed. So ATP is formed. But this is not a direct process that oxygen directly converted into water. No, there are intermediates. So what are the intermediates? There are three intermediates which are formed. The first intermediate which is formed is superoxide ion. The first intermediate which is formed is superoxide ion. The second intermediate which is formed is hydroxyl ion. And the third intermediate which is formed is H2O2 hydrogen peroxide ion. And ultimately it gets converted into water. So these are the three intermediates. So it is formed in all of us. Now let me take my any cell from my head to my toe, any cell of my human body, right? So in my all cells mitochondria are present. Agree or not agree? Yes. In my all cells mitochondria is present. So inside all mitochondria this is taking place. Inside all mitochondria is the powerhouse of the cell. Inside all my mitochondria this is taking place. So you should ask me a question that, ma'am, you are saying it is taking place inside all cells. So you mean the reactive oxygen species are formed in all your cells. So why they are not damaging your cell? You are saying these are reactive oxygen species, no? These are the free radicals. These are formed in all our cells because these are the intermediates. We cannot avoid it. We cannot directly convert oxygen to water. These are the bypass. These are the bypass products. These are the intermediate products. Bypass nahi byproducts. These are the byproducts or intermediate. We cannot avoid that. Yeah, I agree. It is formed inside my all cells. Then why it is not killing my cell? Why it is not causing damage to my cell? These are reactive now. Because as soon as they are generated, the cell eliminates them. Cell has a backup to eliminate them. Cell has a vacuum cleaner to eliminate them. So who is our vacuum cleaner in the cell? In the cell, God has given us three enzymes. What are the name of those enzymes? What are the name of those enzymes? Let me tell you the name of those enzymes. There are three enzymes God has given us. Glutathione peroxidase, number one. Glutathione peroxidase, catalase, the second one, and the third one. I will tell you the three names. So these are the three enzymes which are present in the cytoplasm that will cause elimination of this. Give me a thumbs up if you are getting it. So see, what are free radicals? What are free radicals? See this diagram. Can you see this is oxygen? It is getting converted into water in the mitochondria. This is the process of oxidative phosphorylation. How it get converted? Beech mein teen intermediate. What are those? The first is superoxide, O2 negative. The first is superoxide. Second is H2O2, hydrogen peroxide. And the third one is OH negative, hydroxyl ion. These three are the free radicals, right? And ultimately it get converted into water, right? It is giving 1 electron, 2 electron, 3 electron, 4 electron. Ultimately, because of the reaction of oxygen to water involves 4 electron donation. It is doing 1 electron donation, then 2 electron donation, 3 electron donation. Ultimately 4 electrons are donated and water is formed. Everyone give me a thumbs up. You have studied this in biochemistry. Come on, come on, say yes or no. Say yes or no. So see the last step. See the conversion of H2O2 into OH negative. How it is converting. How it is converting. This conversion takes place by two mechanisms. If this conversion takes place in presence of iron, Fe, iron. If it takes place in presence, it is known as Fenton reaction. It is known as Fenton reaction. Give me a thumbs up. It is known as Fenton reaction. So what is Fenton reaction? So in the Fenton reaction, H2O2 get converted into OH negative in presence of iron. In presence of iron. Have you got it? And if this conversion takes place, instead of iron, it takes place in presence of oxygen. Instead of iron, can you see it is oxygen, superoxide and oxygen. It is known as Weber-Hess reaction. So what is Weber-Hess? So same conversion. H2O2 get converted into OH negative. In both of them, H2O2 get converted into OH negative. But in presence of what? If it takes place in presence of iron, it is known as Fenton, F for F. And if it takes place in presence of oxygen, it is known as Weber-Hess. Give me a thumbs up. Give me a thumbs up. I'm trying hard. Give me a thumbs up. Have you got it? So let me summarize till now. Okay, I will summarize in the end. Let me go ahead. So there are three types of free radical. What are the three types of free radical? Superoxide ion, O2 negative, hydrogen peroxide, H2O2, and hydroxyl ion, OH negative. These are the three free radical. All of them are formed in the mitochondria as the intermediates of oxidative phosphorylation. While converting oxygen to water, these three are formed one by one. Have you got it? There are other two free radicals also. That is nitric oxide and halide. That is chloride, bromide. These are also there, but rarely they are present in the cell. But you should know there are other also. Apart from these three, there are few other free radicals also. Sometimes they are also formed. What it causes? The free radicals are very reactive. They are very reactive. They damage protein, lipid, carbohydrate and nucleic acid. So cell में होता क्या इन चार के अलावा? Cell have only 4 components ना. All 4 are damaged. So cell is damaged completely. Number one. Have you got it? So you can see a cell. So there is lipid damage, there is protein damage, there is DNA damage, there is cytoskeletal damage. So these free radicals are causing everything damage in the cell. Give me a thumbs up. Give me a thumbs up. For their removal or their elimination, God has given us three enzymes, the three backup, the three enzymes. What is the name of the enzyme? It is superoxide dismutase. The first enzyme, glutathione peroxidase. The second enzyme, and catalase, the third enzyme. So these are the three enzymes which eliminate them. Give me a thumbs up. Apart from it, there are few vitamins, vitamin C and E, also eliminated, and transferrin, ferritin, ceruloplasmin, they also eliminated. So these are the removal. For the removal, we use them. Give me a thumbs up. So see the complete equation. I will also write it for you. See everyone. So this is, you will tell me, this is oxygen. I want to convert oxygen into water. How do I convert? बीच में intermediates क्या क्या हैं? So first is O2 negative, then H2O2, learn in sequence only, and then OH negative. Have you got it? Give me a thumbs up if you got it, everyone. So this is the complete sequence. These three are the intermediates. So this reaction is more important. Let me tell you this reaction. H2O2 to OH conversion, if it takes place in presence of iron or takes place in presence of oxygen. If it takes place in presence of iron, it is known as Fenton reaction. If it takes place in presence of oxygen, it is known as Weber-Hess reaction. Give me a thumbs up. Give me a thumbs up. Now I want to eliminate them. I want to eliminate them. So God has given us three enzymes to eliminate them. What is the name of those enzymes? So let me tell you the mechanism of action of these three enzymes. See here, the first enzyme is the superoxide is less toxic. H2O2 usse zyada toxic है, and the most toxic is hydrogen peroxide, hydroxyl ion, OH negative. So I want to convert them ultimately into the water. I want to convert everything into the water, right? So the first enzyme, superoxide dismutase, convert O2 negative to H2O2. That is the first enzyme. That is the first enzyme. Have you got it? The second enzyme, glutathione peroxidase, convert H2O2 to OH negative, right? And the third enzyme, catalase, directly convert H2O2 into water. I cannot simplify more than this. Now everyone give me a thumbs up. Everyone give me a thumbs up. See this diagram. This is the summary. This is the summary. So Preeti, you want to know my schedule. So Preeti, I'm an educator on Unacademy. So few of my classes, my free classes I'm talking, few of my free classes are on YouTube, are on YouTube channel of Unacademy. So currently you are attending my class on Let's Crack NEET PG. So my few classes on Let's Crack NEET PG, Unacademy channel of Unacademy. Few are on Unacademy Live, UA Live. There is another YouTube channel of Unacademy only. And third, few classes are on, I'm sorry, what happened? Let me go back. And few classes are on, just a second. It got zoomed. It is not coming again. Does anyone tell me how it will come? I have to do escape. Okay, got it. And some of my classes are on Future Doctors. That is the third Unacademy YouTube channel. So mainly my classes are on YouTube, the free classes. On Unacademy also, I take free classes on the app. So you have to install Unacademy Learners app. So on installing the app, you have to select NEET PG category and search my name. My name is Dr. Priyanka Sachdev. Search my name in Unacademy Learners app. You will find my name, and along with my name, you will find my profile link. I suggest you to follow my profile link. You will get all the details there. I'm having more than 500 recordings of the free lectures of multiple topics of patho, pharma, micro, medicine, multiple topics. All are available for free along with the PDF of the notes. You can download the notes also. The only thing you will require a code to unlock it. The code is my surname Sachdev 10. Note down the code. It is SACHDEV. Sachdev 10 is the code to unlock any free recording on the app. On the app, it is available. Multiple recordings are available. So please distribute this code to everyone on your batch groups, to your seniors, juniors, interns, all the medicos you know throughout the world, throughout the globe. Distribute this code so everyone can get benefit of the free knowledge. Right? Okay. So should I proceed ahead? Should I proceed ahead? Biochemistry. Shriyansh, definitely I will try to teach you biochemistry also. Anyways, let me finish this. So the three enzymes I have taught you. You can see the same thing. It is written here. It is written here. So see, it is in the free radicals. You see, this is the first thing. Superoxide ion get converted into H2O2, and H2O2 get converted into OH negative. And OH negative ultimately get converted into water. So the first enzyme, superoxide dismutase, helps in this conversion. Glutathione peroxidase helps in this conversion, and catalase directly converts H2O2 into water. Give me a thumbs up. Give me a thumbs up if you got it. So the first enzyme, superoxide dismutase, right? It converts it converts superoxide ion into H2O2. As I have told you, superoxide to H2O2 conversion takes place by superoxide dismutase. The second, glutathione peroxidase. It converts the second thing, H2O2 into OH negative. It is converted by glutathione peroxidase. And the third one is catalase. Can you see this one is catalase? It is converting H2O2 directly in water. Let me write it. I guess you will be more comfortable here. So this is oxygen. It will get converted into water by formation of three intermediates. What are the three intermediates? The first is superoxide ion, then H2O2 ion, and the third one is OH negative ion. Give me a thumbs up. Fatafat, give me a thumbs up. You can see this conversion. Everyone give me a thumbs up. Everyone. So what all enzymes are there for their removal? There are three enzymes. The first is superoxide dismutase, which cause conversion of O2 into H2O2. The second is glutathione peroxidase, which cause conversion of H2O2 into OH negative. And the third one is catalase. This one is catalase, which directly convert H2O2 into water. It directly convert H2O2 into water. Everyone give me a thumbs up. It is directly converting water. So three types of enzymes are present. You have to learn the name of the enzyme, SOD, GP, and C, and what conversion actually they are doing. They are participating in free radicals. So this is the summary of the free radicals in front of you. So everything about the free radical is written in front of you. These all free radicals are formed in inner mitochondrial membrane. Right. One more thing which I have enumerated here. What is that? Yeah, this conversion is important. If it takes place in presence of iron, it is Panton's reaction. If it takes place in presence of oxygen, it is known as Weber-Hess. So have you got the summary? Should I proceed ahead? Right. So that is the thing. So the summary is in front of you. See the summary. Everyone see the summary. It is superoxide get converted into H2O2 and H2O2 get converted into hydroxide. So this is the conversion. You can see the conversion. Give me a thumbs up. I am done with free radical injury. You want me to revise all five or it is done? You want me to revise all five? So there are five mechanisms of cell injury: ATP depletion, mitochondrial damage, loss of calcium homeostasis, defects in membrane permeability, and free radical. I will try to revise. Take an empty page and start writing with me. The first mechanism is ATP depletion, because of which there are three deleterious effects on the cell. The second is mitochondrial damage. Again, it is having three deleterious effects on the cell. The third is calcium homeostasis, the loss of calcium homeostasis. It will lead to activation of four enzymes. I will tell you the name of the enzymes. The fourth is membrane damage. I will talk about the three membrane: plasma membrane, mitochondrial membrane, and lysosomal membrane. And last is free radical. There are three types of free radicals I have already enumerated. Everyone give me a thumbs up. I will try to fill it for you. I will try, right? So you have to help me. So ATP depletion, what are the three mechanisms? Who will tell me the three mechanisms? Due to ATP depletion in the cell, due to ischemia, ATP is not formed. If ATP is not formed, sodium potassium pump will not function. Sodium potassium pump will not function. So more and more sodium inside the cell, more and more water inside the cell. That will cause cell swelling and cell blasts. Give me a thumbs up. Second, if ATP is not formed, ribosome will detach from the endoplasmic reticulum. No protein formation. Protein synthesis will not take place in the cell. The third, anaerobic glycolysis will take place. Due to anaerobic glycolysis, lactic acid is formed that will reduce the pH of the cell and that will cause clumping of the nucleus. The nucleus will get clumped. Everyone give me a thumbs up. Those who are attending the lecture from the beginning, they know what I mean. Everyone give me a thumbs up. The second mechanism, due to mitochondrial damage, three things will happen. Normal oxidative phosphorylation will not take place and abnormal oxidative phosphorylation will take place. Normal में oxygen get converted to ATP, so ATP will not form, ATP depletion, and abnormal में oxygen get converted into ROS, that is free radicals. So free radicals are formed that will damage the cell, and cytochrome C will leak out that will cause apoptosis. So that is the summary of mitochondrial damage. Give me a thumbs up. Due to calcium homeostasis, the imbalance in calcium homeostasis. So calcium is normally present extracellularly and very rarely it is present intracellular in the mitochondria and endoplasmic reticulum. Due to injury, the calcium will leak in the cytoplasm, extracellular also, intracellular also. There is abundant of calcium in the cytoplasm. That abundant of calcium in the cytoplasm causes activation of four enzymes. What is the name of the four enzymes? It is endonuclease, phospholipase, protease, and ATPase. Right? So ATPase will degrade ATP, protease will degrade protein, phospholipase will degrade phospholipid, that is cell membrane, and endonuclease will degrade the DNA. Everyone give me a thumbs up. So the cell will be injured. That is the third mechanism. Fourth mechanism, I will talk about three cell membranes. What are the three cell membranes here? Plasma membrane. Plasma membrane में hole होंगे, so everything will leak out, cell will die. Mitochondrial membrane. I have already explained you here. mitochondrial membrane, the 3 deleterious effects. And lysosomal membrane. Lysosomal membrane is the suicide bag. The cell will commit the suicide because the enzymes will leak out. Give me a thumbs up. Come on. And last free radical just now we have done. We know the names of the 3 free radicals. That is O2 negative, H2O2, and OH negative. How they are formed, how they are converted. We know the name of the 3 enzymes also. SOD, superoxide dismutase, glutathione peroxidase, and catalase, which helps in eliminating these free radicals. Everyone means everyone. Give me a thumbs up. No one in this world will super simplify more than this. I can challenge. So you want me to launch some MCQs based on these mechanisms, which are already asked in your previous year question papers in NEET PG, FNG, and ICT? If yes, give me a thumbs up. So this is the first question in front of you. I want you to write the answer in the comment box. I want you to write the answer in the comment box. So everyone, please everyone, Zishan, Vasi, Preeti, Priyansh, everyone, those others who are also attending, I don't know the names, Nibu, I want everyone to participate. What is your answer from ABCD? Write down what is your answer, ABCD, in the chat box. So let me read the question. The question is in front of you. Deadliest free radical. Who is the most deadly free radical? Yes, so Sohil is saying B. Who is the deadliest? So H2O2 is also a free radical, O2 negative is also a free radical, and OH negative. This one is not a free radical. But from the first 3, which is the deadliest? Correct answer is C, Vasi. You are absolutely right. So I have already taught you in the lecture that OH negative is the most deadliest. So first O2 negative is formed. It is not very dangerous. It converted into H2O2. It is not also very dangerous. The most dangerous is OH negative. That is the most dangerous, most deadliest. So the correct answer is C. Give me a thumbs up. This is the next reaction. Define Fenton reaction. What is Fenton reaction? In Fenton reaction, what is get converted to what? You can read the 4 options are in front of you. What is Fenton's reaction? So radiant energy is absorbed by the water. Hydrogen peroxide is formed by myeloperoxidase. Ferrous ion get converted to ferric ion. Or nitric oxide get converted into peroxynitrate ion. What is the correct answer? Yes. What is the correct answer? I am asking to you. Yes. Good. The correct answer here is B. So it is in presence of iron. In presence of iron, H2O2 get converted into OH negative. Right? In presence of iron. So actually, iron may, the ferrous iron get converted into ferric iron during this process. That is in presence of iron. Yes, C. Very good. Everyone, Vasi, Preeti, Sohil, Zishan, you all are right. The correct answer is C. Very good. This is the next question in front of you. I guess everyone will be right in this question. Everyone will be right in this question. Right? Which of the following is not a free radical scavenger? I'm asking scavenger. Scavenger are the enzymes which eliminate the free radical. So which of the following is not a free radical scavenger? Please tell me the name. Is it glutathione peroxidase, superoxide dismutase, catalase, or xanthine oxidase? Yes, Sohil, Zishan, you are absolutely right. Xanthine oxidase is not the name of any enzyme. But the other 3 are the enzymes which causes the elimination of free radicals. So yes, the correct answer is D. This is the next question in front of you. Enzyme that protects the brain from free radical injury. Tell me the enzyme which protects not only brain, the whole body from free radical injury. Name the enzyme. There are 3 enzymes now. There are 3 enzymes. Is it myeloperoxidase? Is it superoxide dismutase? Is it MAO? That is monoamine oxidase, or is it hydroxylase? So what is the correct answer? Yes. So the 3 enzymes are superoxide dismutase, glutathione peroxidase, and catalase. Out of the 3, one of them is given in the option. So correct answer here is B. Yes, Zishan. The correct answer here is B. Yes, very good. Have you got it? Yes, Vasi. The correct answer is B. Very good. So B is the correct answer. Should I proceed? This is the next question in front of you. Okay, leave this one. So, okay. So I have taught you the various mechanisms of the cell injury. In the next lecture, I'm going to teach you the morphology of the cell injury along with the cell death, that is apoptosis and necrosis. So today I'm going to take 3 more lectures in the evening today, right? So thank you very much for being with me. I would like to stop here right now. I would like to continue the ahead portion in the next lecture, which I will take in the evening. I request you to join me in the evening and the night. 3 more lectures today. Thank you very much for being with me. I really appreciate your time that you have given to me. If you have liked the lecture, don't forget to click on the like button before you leave the lecture. Please click on the like button before and share the link with your all friends, colleagues. Please subscribe our YouTube channel, all YouTube channels of Unacademy NEET PG, that is Let's Track NEET PG, Future Doctors, and UA Live. And don't forget to click on the bell icon for the notifications. On Unacademy, we are launching 3 new offerings. if you take the subscription, the paid subscription of Unacademy, you will get previous year question bank of NEET PG, FMG, and INICT that you can do the practice with details explanation. We are launching a doubt clarification batch. So even if you have the slightest doubt, you can ask to your favorite educator on the app. So for that, you have to take the subscription, and practice test after every plus class. After every class, we will give you a test of 5 to 10 questions based on that class. So topic by topic, you will be get prepared for that, right? So these are the question bank, doubt clearing series. This is daily practice papers. Now we are launching Prof 1. Prof 1, those all students who are in Prof 1 or friends are in Prof 1, please inform them. So we are launching Prof 1, your path towards cracking MBBS and next. So in that, we will help you in cracking the university exam of MBBS with distinction along with the next preparation. So I request the students, the first year students, to join this batch, take the subscription and take the benefit from the topmost educators. Once you take the subscription, there are two types of subscription on Unacademy. Plus, if you take the plus, you will get access to Unacademy. If you take the iconic, you will take access to Unacademy and prep ladder. So according to me, you should go with iconic. The price difference is very less, but here additionally you are getting prep ladder also, right? So once you take the subscription, you will be eligible for these all batches from the topmost educators of the India. We all work as a team, right? So these are the various plans of the plus subscription. These are the plans. Iconic subscription, these are the plans. And light subscription, you will get this hard copy of the notes. Hard copy of the notes also in this subscription. So these are the plans. So whatever subscription you want to take, you can see minimum 2 months, maximum 4 years. Longer the subscription, cheaper it is. So whatever subscription of your choice, of your wish, you can take it. Before taking any subscription, please apply the code Sachdev10 before payment. You will get straightforward 10% discount if you apply this code. So the code is useful. I guess the code is useful for all students. If you apply the code Sachdev10 on the app to unlock the free classes, if you want to watch only free classes, right? If you want to take the subscription, the paid classes, you will get discount with the same code. So I request you to distribute the code on your batch group, seniors, juniors, interns, to all the medicos throughout the world. Please use this code to take the subscriptions of the Unacademy for getting 10% straightforward discount, authentic discount, and to unlock the free classes. So thank you very much. Study hard. Bye-bye. I'm ending this lecture, and